Now whatever remains at the top in the cheesecloth and which doesn't get filtered, that we call it as the acid insoluble pool, and it is called as the retentate. So coenzyme means there are some enzymes which cannot function on its own. At that time a coenzyme needs to be added, wherein that will go and bind with the enzyme and help in increasing the speed of the reaction. Replica means producing a copy of something. So here a copy of the DNA is produced. It's called as DNA replication. Hello everyone, a warm welcome to the session of revision class for first PUC Biology. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session of chapter Biomolecules, we will learn about the synopsis, that is what and all is there in this particular chapter. We'll have a look into it and then move on to learning some of the important multiple choice questions under this particular chapter. So one multiple choice question will be asked from this particular chapter. One 2-mark question will be asked and one 3-mark question will be asked. So therefore, totally this chapter carries a total weightage of 6 marks according to the blueprint given by the PU board. So in this particular session, we'll learn about what and all are the important contents under that particular chapter in the form of a chart, and then we'll move on to learning about the multiple choice questions. So 2 marks and 3 marks, we will learn it in the next session of this particular chapter. In this chapter, the first topic that is there is analysis of chemical composition. So any plant or any animal material, you need to find out what compound is there in that particular plant and animal material. You need to go in for some biochemical essays, right? So for that biochemical essay, you need to get the material. So you collect the plant material, you collect the animal material, that is the tissues, you take them and you grind it. So how it is done is what we see. So be it the plant or the animal material, whatever it is, first you take a piece of the material according to the grams or the amount, the weight that is mentioned. You take it in a mortar and a pestle, neatly you grind it. So while grinding, you add trichloroacetic acid to it. After adding the trichloroacetic acid and grinding it into a fine paste, you take a cheesecloth or a muslin cloth and then you strain out. So when you strain, you'll get 2 fractions. One is the filtrate and other one is the substrate. So what do we do here? We go in for, there are 2 types of analysis of chemical compounds. One is chemical analysis and the other one is ash analysis. So whatever I told you now comes under chemical analysis. So what is done here? The tissue is ground using a mortar and a pestle with addition of trichloroacetic acid. And then whatever material is ground, that fine paste is taken and it is filtered through a cheesecloth. Now, once you filter it through a cheesecloth, you'll have a liquid that is there collected in a container or in a beaker, and you have that is retentant, that is the solid matter that is there. Now, therefore, we call it as the acid soluble pool and the acid insoluble pool. So, whenever you filter, whatever collects, that liquid, whatever collects at the bottom, that will be the acid soluble pool and it is called as the filtrate. Now, whatever remains at the top in the cheesecloth and which doesn't get filtered, that we call it as the acid insoluble pool and it is called as the retentate. Now, whatever is there in the acid soluble pool or the filtrate, they are the biomicromolecules. So, biomicromolecules, micro means smaller in size, and they are smaller in size and therefore they are capable of passing through the cheesecloth and settling as a filtrate. Now, whatever is left over, acid insoluble pool or in the cheesecloth, we call it as the retentate, and that retentate consists of biomacromolecules, and these biomacromolecules are larger in size, that is more than 10,000 Dalton. They are larger in size and therefore they cannot pass through the cheesecloth and therefore they remain in the cheesecloth as retentate itself. So each of these we will study separately later. Now we got to know how chemical analysis is done. Now moving to ash analysis. In ash analysis, there is no grinding and all that. You take a plant tissue or you take an animal tissue, dry it under the shade drying, you do it, and then you burn the dry leaves or dry tissue. It is burned. and whatever ash you got, that is again subjected to further analysis to find out what compounds are exactly present in it. That is about ash analysis. Now moving back to here, I told you in chemical analysis, the end products that we get is biomicromolecules and biomacromolecules. That is biomicromolecules in the filtrate and biomacromolecules in the retentate. Now what are there? What are the different types of biomicromolecules and different types of biomacromolecules we shall look into? So biomicromolecules, I told you their size is very, very small, meaning their molecular weight is just 18 to 800 Dalton. Therefore, they are capable of passing through the cheesecloth and getting filtered. Now, those biomicromolecules are of different types. One is amino acids, lipids, and the other one is the heterocyclic compounds. Now, here lipids, though they have a molecular weight which is less than 1000 Dalton, but still the lipids doesn't get filtered through the cheesecloth. That is one of the important characteristic feature of the lipid, because lipids are not soluble in any of the compounds. So that is why they do not get filtered. Now moving further, we'll study about each of the biomicromolecule, that is amino acids. So what are the different types of amino acids and what is the structure or forms of amino acids? So talking about the types of amino acids can be acidic amino acids, basic amino acids, neutral amino acids and aromatic amino acids. So acidic amino acid, one of the best example is glutamic acid. For basic amino acid, it is lysine. For neutral amino acid, it is valine. For aromatic amino acids, it's tryptophan and tyrosine. There are 20 to 21 different types of amino acids, but I have put here the examples that have been mentioned in the NCERT textbook. Next, moving to the structure and forms. Based on the structure and the forms, the amino acids can exist in protonated form. Protonated form means there is an addition of the H plus ions, Zwitter ionic form and deprotonated form. So protonated form, the formula is H3N, that is NH3 plus CHRCOOH, wherein this R is the variable group, and here as you can see there is addition of the proton here, so therefore it is protonated. Talking about the Zwitter ionic form means they can exist in two forms, that is anionic form and cationic form, wherein they carry a positive as well as a negative charge. So that is a Zwitter ion form. Next is deprotonated, they will be losing a H plus ion, therefore they will carry a negative charge. So these are the different structures of amino acids based on the charge or the protons they carry. Next, moving further, lipids. So lipids are of different types: fatty acids, glycerol, esters of fatty acids with glycerol, phospholipids and cholesterol. So fatty acids are again of two types: saturated fatty acids and unsaturated fatty acids. So saturated fatty acids means between the carbon chain, they do not have a double bond. So entire carbon, so if 10 chains of carbon atoms are there in a particular fatty acid and there is no double bond at all in between, then we call it as a saturated fatty acid. Now unsaturated fatty acids means between the carbon atom, there lies a double bond. Either the double bond can be between one or two carbon atoms and more than that also, but if anywhere, if between a carbon atom in the chains of fatty acid chain, if you find a double bond in between, then it is called as the unsaturated fatty acids. And one more thing about saturated and unsaturated fatty acid is, so that is about saturated and unsaturated fatty acids. Moving to esters of fatty acids with glycerol. These are the fatty acids which are bonded to a glycerol or glyceride. So here, depending on that monoglyceride, they'll have a single glyceride. Diglycerides 2, triglycerides are 3 glyceride compounds. Now in triglycerides, there are fats and oils. So fats usually have a high melting point, for example butter, and oils usually have a low melting point, for example gingerly oil. Next, moving further, heterocyclic compounds. So heterocyclic compounds means under this the nitrogenous bases, the nucleosides and the nucleotides will come. So nitrogenous bases are nothing but carbon compounds which have a heterocyclic ring, and they usually a 6 carbon compound having or their carbon compounds having a heterocyclic ring, they are again divided into purines and pyrimidines. So in purines and pyrimidines, under purines there is adenine and guanine. Under pyrimidine there is cytosine, thymine and uracil. Adenine, guanine, cytosine and thymine, you get to find them in DNA. Adenine, guanine, cytosine and uracil, you get to find them in RNA. Now this is the structure of adenine. This is the best example wherein we can say it is made up of two heterocyclic rings and they have the carbon compounds, and this is the structure of the adenine. Now and this is the structure of uracil. Next, moving further, talking about the nucleosides. Nucleosides means they'll have a nitrogen base. For example, it can be adenine, guanine, cytosine, thymine or uracil, which is connected to a phosphate sugar molecule. That can be the phosphate sugar which is connected to a sugar molecule. That sugar can be a ribose sugar or a deoxyribose sugar. If it is a RNA, it will be a ribose sugar. If it is a DNA, it will be a deoxyribose sugar. Now here, based on that, it is adenosine, guanosine, cytidine, thymidine and uridine. So this is the structure of adenosine. So what happens? This adenine structure that is there, that will be here, and this adenine, that is the nitrogenous base, it will be connected to a sugar. Now this is a sugar. This sugar can either be a deoxyribose sugar or a ribose sugar. Next, moving further, and this is about uridine, same also, it is also the same thing, but here uracil is connected to this will be a ribose sugar. Why? Because we don't get uracil, it is not there in DNA. So therefore, it is there only in RNA. So RNAs are always having a ribose sugar. Now this then adenine, this can be a ribose or a deoxyribose. Next, moving further, nucleotides. Now what is a nucleotide? Nucleotide means nucleoside attached to a phosphate group. Now what is this nucleoside? A nitrogenous base that is attached to a sugar molecule, be it ribose or deoxyribose, and that is attached to a phosphate group, that is PO4 group, phosphate group. We call it as PO4 group, we call it as nucleotide. So these two, this is very, very important. Nucleoside and nucleotide, they are very important. So examples are adelinic acid, guanylic acid, cytidilic acid, thymidilic acid and uridylic acid. So this is an adenilic acid wherein we have adenine, which is a nitrogen base that is connected to a sugar and that is connected to a phosphate group. So altogether, we call it as a nucleotide. Next, moving to biomacromolecules. So biomacromolecules usually have a size of about 10,000 Dalton and they are of different types. Proteins, polysaccharides, nucleic acids, all of them come under the biomacromolecules, meaning they are the written tense. They are present in the acid insoluble pool. Now, proteins are of different types based on their structure. It can be primary protein, secondary protein, tertiary protein and quaternary protein. And the adult human hemoglobin is one of the best example for quaternary protein. Next, talking about the functions of protein. Proteins help in the transport of nutrients across the cell membrane. They help, some of the proteins act as antibodies wherein they help in fighting infectious organisms. Some of the proteins act as hormones which carry messages from one part of the body to the other, and some of the proteins act as enzymes which are needed for all the metabolic activities to take place in a body. Next, talking about polysaccharides. Polysaccharides are of two types, homopolysaccharides and heteropolysaccharides. So what do we mean by polysaccharides? Saccharide means sugar, poly means many, mono means single. In that, okay, poly means many, mono means single. In here we have homopolysaccharides and heteropolysaccharides. So homo means similar, hetero means different. So those polysaccharides, those carbohydrates in which they are connected by same saccharide units are called as homopolysaccharides, and those in which the saccharide units are not the same, we call it as a heteropolysaccharide. So for homopolysaccharide, the example is starch, cellulose, inulin and glycogen. For heteropolysaccharide, lignin is the best example. Next, moving to enzymes. So enzymes are those compounds which catalyze the rate of a reaction, or they are capable of speeding up the rate of a reaction. Now, enzymes are of different classes and types. So here, different classes of enzymes. We have oxidoreductases, transferases, hydrolases, lyases, ligases and isomerases, which have their own specific function in bringing about the or increasing the rate of the reaction. Now, types of enzymes, it is of simple enzymes and conjugate enzymes. So, conjugate enzymes means those enzymes which function only when it is attached to another enzyme molecule. Say for example, we have coenzyme. So NAD and NADP plus are the two important coenzymes. So coenzyme means there are some enzymes which cannot function on its own. At that time, a coenzyme needs to be added, wherein that will go and bind with the enzyme and help in increasing the speed of the reaction. So that is called as coenzyme. Now, prosthetic group is nothing but heme. Then it consists of metal ions. So, say for example, some of the enzymes cannot function on its own. It needs a metal ion. Say for example, zinc, Zn2 plus, that is in carbonic anhydrase. So carbonic anhydrase enzyme can speed up the reaction process, wherein, say for example, you add carbon dioxide and water to get carbonic acid. You need to add an enzyme, carbonic anhydrase. Now, adding carbonic anhydrase itself is not sufficient. There, metal ions needs to be present also if the speed of the reaction has to increase. So therefore, for metal ions, this is one of the best example. Next, factors affecting enzyme action. Temperature is one of the important factor. Any change in the temperature, slight increase in the temperature can denature the enzyme, because at the end of the day, enzymes are nothing but proteins. Next, pH. So, certain enzymes only act at a particular pH. And substrate concentration. The amount of substrate that are used to bring about a reaction also affects the rate of the enzyme activity. Next, there are some inhibitors. Inhibitors are the ones which prevent the action of the enzyme. Under that, two types are there: competitive inhibitors and non-competitive inhibitors. So, competitive inhibitors. So, we all know that in enzymes, they have an active site wherein the substrate needs to go and bind in that active site, right? So, what happens in the case of competitive inhibitors is that these competitive inhibitors will go and bind in that active site. So, when it is already bound to that active site, there is no space for the substrate to go and bind, therefore halting the reaction process. So, that is called as competitive inhibitors. Now, non-competitive inhibitors means these non-competitive inhibitors will not go and bind to the active site, but it will go and bind somewhere else in the enzyme, therefore altering the shape of the enzyme. So, when the shape of the enzyme is altered, the substrate cannot go and bind to the active site even though it is free. So, such inhibitors are called as non-competitive inhibitors. So, this was about an idea of what and all is there in the chapter, we just brushed up. So, we'll move on to the multiple choice questions. So, under this, the first one, which functional groups are present in amino acids? Hydrogen and carboxyl group, amino and carboxyl group, hydrogen and amino group, amino carboxyl and R group. So, it is option D, amino carboxyl and R group, because we know that in an amino acid, it consists of an alpha carbon atom to which on either ends the amino group and the carboxyl group is attached, and to that a variable group or a R group is attached. So, therefore, option D is the right answer here. Next, what is the name given to amino acids with the hydrogen as the R group? So, what is the name given to amino acids with hydrogen as the R group? So, in the variable group is hydrogen is there, it is glycine. Now, for alanine, it is CH3. For glutamic acid, for alanine, it is CH3. For serine, it is CH2OH. CH2OH. So, the question can be asked, what is the name given to the amino acid which is with a CH3 as the R group? What is the name given to the amino acid with a CH2OH as the R group? Because those are the ones that are given in the textbook. So, that is why. So, here the question that I have asked is, what is the name given to the amino acid with the hydrogen as the R group? The answer is glycine. So, in the R group, if hydrogen is attached in the variable group, then it is glycine. Now, how many types of amino acids occur in proteins? It is about 20. So, 20 different types of amino acids are held together by peptide bonds forming a long chain, and that is nothing but a protein. So, proteins consist of 20 types of amino acids. So, next question, which type of amino acid has an ionizable nature for both NH2 and COOH groups? So it is neutral amino acids. So neutral amino acids, if you remember, Zwitter ion. So Zwitter ion exists when the pH is in neutral, that is in 7. So therefore, they have an ionizable nature for both NH2 as well as COOH groups. Next question, what is the variable group designated as an amino acid? It is the R group, because R group is a variable group. Next, which type of amino acid is represented by tyrosine, phenylalanine and tryptophan? So which type? So tyrosine, phenylalanine and tryptophan, they are aromatic amino acids. They come under the aromatic amino acids. So which type of amino acid has acidic nature? It is glutamic acid. And lysine, it is basic. Valine is neutral. And tyrosine, they are nothing but aromatic. Aromatic. So here, if the question comes which type of amino acid as in basic nature, we can write lysine. Which type of amino acid as a neutral nature, we can write valine. Which type of amino acid is aromatic in nature? It is tyrosine or phenylalanine or tryptophan, all are aromatic itself. So the question that I've asked here is which type of amino acid has an acidic nature? So the answer is glutamic acid. Moving further, what is the characteristic of lipids? Generally water soluble, generally water insoluble, both A and B, none of the above. It is generally water insoluble. Lipids are insoluble in water because fats, we all know that you put fats in water, will it get like sugar and salt, will it get soluble in water? No, right? So therefore, they are generally water insoluble. Next, what is fatty acid composed of? Hydroxyl group, carboxyl group and R group, both A and B, none of the above. So it is carboxyl group and R group. So fatty acids are composed of carboxyl group and R group. Next question. So what could be the R group in a fatty acid? It can be CH3 group, C2H5, both A and B or higher number of CH2 groups. It is both A and B, that is it can have CH3, the R group can be a methyl or it can be a ethyl, that is a CH3 and a C2H5. So therefore answer is both A and B. So what is the number of carbon atoms in which one? Palmitic acid. So it is 16. Now, if they ask the question, what is the number of carbon atoms in arachidonic acid, then it is 20. For arachidonic, it is 20. But here the question that I've put is for carbon atoms in palmitic acid, it is 16. That is, palmitic acid is nothing but a fatty acid which are made up of chains of 16 carbon atoms. Next question, which simple lipid is composed of trihydroxypropane? So next question, which simple lipid is composed of trihydroxypropane? So trihydroxypropane, we find it in glycerol. Next question, what is a characteristic of oils? They have higher melting point, they have a lower melting point, they solidify in winters, none of the above. That is, they have a higher melting point and they solidify in winter is for fats. It's not for oils. These 2 points that are there, it is for fats. Now for oils, it is they have a lower melting point. So that is why oils, even in winter also, they'll remain in the liquid form itself. They don't solidify. So option B is the right answer here. Next question. What are lipids with phosphorus and a phosphorylated organic compound called? So the name itself says phosphorus, phosphorylated. They are called phospholipids. And where do we find these phospholipids? In the cell membrane. Hope you remember in the previous chapter. Next question. What is the name of the nitrogen base found in DNA that pairs with thymine? So we have to find out that pairs with thymine. What pairs with thymine? Adenine. What pairs with guanine? Cytosine. What pairs with adenine? Thymine. What pairs with cytosine? Guanine. Uracil you cannot consider here because we are talking about DNA. Because in DNA uracil is not there. Next question. When a sugar is attached to a nitrogen base, it is called as, what is it? Sugar is attached to a nitrogen base, it is called as nucleoside. Now don't get confused. How you can remember is, see there is a S here, no? So always remember, sugar will be present. Sugar is attached to nitrogenous base. Right answer here is nucleoside. Nucleotide is for nucleoside attached to a phosphate group. That is nucleotide. Next question. Which of the following is not a nitrogen base found in DNA? What is it? Not a nitrogen base. To concentrate on this. Not a nitrogen base found where? In DNA. Is it adenine, cytosine, guanine, uracil? It is uracil because uracil we find it only in RNA. Next, what are nucleic acids made of? Nucleosides, sugar molecules, nucleotides, amino acids. What are nucleic acids made up of? The right answer is nucleotides because we have nucleic acids that are present in the DNA wherein it consists of a sugar molecule which is attached to a nitrogenous base and a phosphate group. So therefore it is made up of a nucleotide. Next question. So which of the following is a nucleotide? It is adenylic acid. So adenosine, thymidine, guanosine, all of these are nucleosides. So they are nucleosides, but what I've asked here for nucleotide. So adenylic acid is a nucleotide. Next question. How do DNA and RNA differ? DNA contains the nitrogen base uracil while RNA contains thymine. No, it is wrong because DNA contains thymine, RNA contains uracil. That is how it has to be. This should actually come here and this should actually go here. So that is why it's wrong. Next one, DNA is double-stranded while RNA is single-stranded. DNA contains deoxyribose sugar while RNA contains ribose sugar. All of the above. So we can say DNA contains deoxyribose sugar while RNA contains ribose sugar. So that is the right answer for this. Next question, what is the name of the process by which DNA is copied during division? Is it called replication, transcription, translation, reproduction? It is called replication. Replica means producing a copy of something. So here a copy of the DNA is produced. It's called as DNA replication. Next question, which nitrogen base pairs with cytosine in DNA? So cytosine, it is guanine. Because we know that adenine base pairs with thymine with a double hydrogen bond. If it is a DNA, and adenine base pairs with uracil if it is a RNA, and guanine with a triple hydrogen bond, it base pairs with cytosine if it is a DNA or a RNA. So this is how it is. So next question, what is the molecular weight range of compounds found in the acid soluble pool? So acid soluble pool means it is the filtrate, that filter that you get at the bottom, it's the filtrate. Now they are biomicromolecules, meaning they'll have a small molecular weight. So what is it? It is about 18 to 800 Dalton. It is about 18 to 800 Dalton. So next question, what is the molecular weight range of biomolecules or micromolecules? So molecular weight range of biomolecules or biomicromolecules, it is less than 1000 Dalton. It should be from 18 to 800, meaning it should be less than 1000 Dalton for biomicromolecules. Next question, what is the molecular weight range of macromolecules or biomacromolecules? So they will be larger in size, that is they will be about 10,000 Dalton. Next question, which of the following is not a biomolecule? Proteins, nucleic acids, lipids and glucose. It is proteins. Proteins are not a biomolecule. So question number 26, which of the following is not a biomolecule? Is it proteins, lipids, nucleic acids, or glucose? It is glucose because proteins, lipids and nucleic acids have a molecular weight less than 1000 Dalton, whereas glucose has a molecular weight more than 10,000 Dalton. So that is why. Next question, why do lipids come under the macromolecular fraction even though they have a molecular weight less than 800 Dalton? This is one of the most important questions that keeps on coming because they form polymeric substances, because they are present in the cell membrane and other membranes, because they have a high molecular weight when in the form of cell membrane and other membrane, because they are not soluble in acid. It's because they are present in the cell membrane and easily they don't get detached from the cell membrane and they don't mix with any of the compound so that it can come as the filtrate. So therefore, they remain in the acid insoluble fraction. So option is option B because they are present in the cell membrane and other membranes. Next question, what are proteins made up of? Polypeptides, carbohydrates, lipids, vitamins. They are made up of polypeptides with nothing but chains of amino acids. Next question, what links amino acids in proteins? Of course, it is peptide bond. That is why they are called as polypeptides. Next question, what is a homopolymer? Again, here polymer means chains of different compounds which are held together by bonds. Homo means same. So polymer of one type of amino acids, polymer of different types of amino acids, polymer of lipids, polymers of carbohydrates. So we can say polymer of one type of amino acid is called as homopolymer. Now, which is the most abundant protein in animal world? Collagen, keratin, rubisco, hemoglobin. So, collagen is the most abundant protein. This question is also quite important. The next one is also important. Which is the most abundant protein in the whole of the biosphere? It is rubisco. It is rubisco. Next, what are polysaccharides? Long chains of fats, long chains of sugars, long chain of proteins, long chain of nucleic acids. So, polysaccharides are long chain of sugars. That is, we get to find them in carbohydrates. Next, what is cellulose made up of? Only one type of monosaccharide, only one type of amino acid, only one type of fatty acid, only one type of nucleotide. Now it is understood cellulose is a form of carbohydrate. It is made up of only one type of monosaccharide. Option A. Next question. Which polysaccharide is present as a storehouse of energy in the plant tissues? So, which polysaccharide is present as a storehouse of energy where in plant tissues? It is starch, because we know that whatever glucose is produced during the process of photosynthesis, that excess glucose gets stored in the form of starch, and whenever it is required, it gets converted into glucose and it travels to different parts of the plant body. So, therefore, it is stored in the form of starch. Next question. Next question. What is the reducing end in a polysaccharide chain? It is the right end. So, the right end is called as the right end of the polysaccharide chain is called as the reducing end. Now, remember R. Remember like this. So, right, reduce. Next question. So, which polysaccharide forms helical secondary structure? The right answer here is, it is starch. So, which polysaccharide forms helical secondary structure? It is starch. Here, most of the time, the option inulin has come. Inulin is nothing but a form of fructose. Okay, that is a form of fructose. So, remember that. So, next question. What is the color of starch I2 molecule? So, it is blue. That is why when we add iodine to test for starch, it gives a violet blue color. Next, what are the plant cell walls made up of? Proteins, fats, carbohydrates, nucleic acids. So here, what are the plant cell walls made up of? Usually, we have the habit of writing the cell wall of plant is made up of cellulose, cellulose, cellulose. And what is that cellulose? It is nothing but a monosaccharide. Now, what is that monosaccharide? They come under the carbohydrates. So, therefore, you can mark the answer as carbohydrates, because carbohydrates is nothing but a cellulose, which is a monosaccharide. So, therefore, option C is the right answer here. Next, what is chitin? A complex polysaccharide found in animals, a complex protein found in animals, a complex nucleic acid found in animal, a complex fat found in animal. Now, chitin is something that is found in fungi as well as in animals. Here, fungi, it can't be a fat, so we'll eliminate this option. Now, it is not found in plants at all. It is not there in bacteria also. So, therefore, option A, it is a complex polysaccharide found in animals. We had studied in arthropods, that is insects. Their exoskeleton is made up of chitin, if you remember. So, therefore, it is a complex polysaccharide found in animals. Next question, what is the N-terminal amino acid? The first amino acid, the last amino acid, both the first and the last, none of the above. It is the first amino acid is called the N-terminal amino acid, and the last amino acid is called as the C-terminal. Next, how many subunits does a human hemoglobin consist of? It consists of 4 subunits, that is alpha and beta, 2 alpha and 2 beta. How many subunits of each type constitute the human hemoglobin? I told you it's 2 subunits of alpha type and 2 subunits of beta type, and that is why they have 2 subunits which looks like 4 wools, that is wound wools that are arranged. So therefore it has a quaternary structure. So this was about the important MCQs under this particular chapter. So I hope you found the session quite useful. So we shall meet again in the coming session discussing a new chapter. So see you in the next session. Thank you.